Today's Real Life Orgo is about the chemistry of digestion, and it's brought to you by Cameron Ward, Davis Hensley, Ian McCluckey, and Spencer Lubin.
There are three major macromolecules that are broken down in digestion: carbohydrates, fats, and proteins. Today we're going to explain some of the chemistry behind the breakdowns of these molecules, and hopefully you can learn a thing or two about organic chemistry in the meantime. Carbohydrates, also called saccharides, are organic molecules that are made up of repeating units, each composed of carbon, hydrogen, and oxygen. They are polymers, a class of molecules built from repeating units called monomers, similar to a necklace built from individual beads or a chain built from its links. Carbohydrates are found in vegetables, fruits, dairy products, breads, and sweets. As you can see on the right, glucose, fructose, and sucrose on the bottom are all versions of carbohydrates. While these are all aromatic rings, some are just chains. Glucose is arguably the most important carbohydrate of all. When oxidized in the body in the process called metabolism, glucose produces carbon dioxide, water, and some nitrogen compounds in the process that provides energy which can be used by cells to carry out all sorts of processes. Glucose exists in two conformations, alpha and beta. The two isomers differ only in the direction that the H and OH groups point on carbon 1. As you can see on the images to the right, the alpha glucose is in the cis conformation, while the beta glucose is in the trans conformation. Glucose is broken down in order to provide energy to the body. The first step in the degradation of glucose is glycolysis, which breaks down glucose to pyruvate. The main purpose of glycolysis is the generation of energy in the form of ATP. One molecule of glucose yields 4 ATP, 2 NADH, and 2 pyruvate molecules. ATP can be used directly for energetic purposes. The pyruvate molecules can be further broken down to produce even more ATP, and NADH is also used in this process. Glycolysis involves 10 enzymatic reactions. First, the phosphorylation of glucose at position 6 by hexokinase. Second, the conversion of glucose 6-phosphate to fructose 6-phosphate by phosphohexanose isomerase. Third, the phosphorylation of fructose 6-phosphate to the 1,6-biphosphate by phosphofructokinase. Fourth, the cleavage of fructose 1,6-biphosphate by aldolase. This yields two different products, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Fifth, the isomerization of dihydroxyacetone phosphate to a second molecule of glyceraldehyde 3-phosphate by triose phosphate isomerase. Sixth, the dehydrogenation and concomitant phosphorylation of glyceraldehyde 3-phosphate to 1,3-bisphosphoroglycerate by glyceraldehyde 3-phosphate dehydrogenase. Seventh, the transfer of the 1-phosphate group from 1,3,5-phosphoglycerate to ADP by phosphoglycerate kinase, which yields ATP and 3-phosphoglycerate. Eighth, the isomerization of 3-phosphoglycerate to 2-phosphoglycerate by phosphoglycerate mutase. Ninth, the dehydration of 2-phosphoglycerate to phosphophenylpyruvate by enolase. And finally, the transfer of the phosphate group from phosphophenylpyruvate to ADP by pyruvate kinase to yield a second molecule of ATP.
Another major nutrient of digestion is fats. Oils and fats are composed of triesters of carbon, hydrogen, and oxygen atoms. Esters are formed when an organic acid and an alcohol react. Water is released in this reaction. The digestion of fats begins in the mouth, where short-chain lipids break down into diglycerides because of lingual lipase. Bile, which contains bile salts, is produced in the liver. The bile salts emulsify the lipids inside the small intestine. The fat present in the small intestine stimulates the release of lipase from the pancreas, and bile from the liver enables the breakdown of fats into fatty acids. Once the lipids are broken down into finer droplets, they have more total surface area, which helps the fat digesting enzyme, pancreatic lipase, which can only act on the surface of the lipid. The enzymes in the small intestine are responsible for almost all fat digestion. Pancreatic lipase acts on the lipid and breaks it down, which results in free fatty acids and monoglycerides, the two digested products of lipids. These products are much easier for the small intestine to handle and are easily absorbed out of the digestive tract.
To the right are the structures of triesters, glycerol, and fatty acid. Triesters are formed when a molecule of glycerol, which contains 3 hydroxyl groups, reacts with 3 separate fatty acid molecules. Lastly is the third major nutrient of digestion, proteins. Proteins are broken down by 3 main enzymes, pepsin, trypsin, and chymotrypsin.
All proteins are made up of amino acids. Amino acids consist of an amino group, which you can see in the green on the picture to the left, a carboxyl group, which is in the orange, and an R group, which is in the red box. 20 types of amino acids exist, and each has a different R group. The 20 different amino acids come together in varying orders to form all of the proteins. Proteins are digested and broken down by the hydrolysis of the carbon-nitrogen bond shown through the mechanism shown here. In this mechanism, the oxygen from the water attacks the orange carbon, pushing the carbons that were shared in the double bond between the carbon and oxygen onto the oxygen. The water molecule attached to the carbon then loses an H+, which is then attacked by the nitrogen to form a bond. The nitrogen then takes the electrons from the carbon-nitrogen bond, and simultaneously the electrons on the orange oxygen move down to form another bond with the C. This mechanism breaks down proteins into the products shown below. Because of the globular or fibrous nature of proteins, the digestive enzymes can't reach the chemical bonds between the amino acids until the proteins have been unfolded by a highly acidic gastric juices in the stomach. Once the proteins have been unfolded, the digestive enzymes in the stomach and small intestines attack specific points in the amino chain and break it into smaller pieces called peptides. They then continue breaking them down into individual amino acids. The chemicals inside the stomach are called pepsin, gastric juice, and hydrochloric acid, pictured in the pictures to the right. Hydrochloric acid in the gastric juice converts the pepsinogen into pepsin by cleaving off a stretch of amino acids called a peptide. This reaction requires a very acidic pH, normally ranging between 1 and 3. The acidic environment is needed for this generation and the activity of pepsin. The hydrochloric acid in the stomach generally provides a pH of about 1.5 or 3.5. As you can see in the chart to the right, the gastric acid is made up of mostly water with trace amounts of solid and organic and inorganic molecules. The acid in the stomach causes food proteins to unfold in a process called denaturation. Denaturation exposes the protein's molecular bonds so that pepsin can access them and break them down into smaller fragments. These fragments are called peptides or polypeptides, and this reaction normally needs to happen in a heated environment.
So this wraps up your real-life Orgo session about the digestive system and the chemistry behind it. You learned about the 3 major types of nutrients, carbohydrates, fats, and proteins, along with the mechanisms behind them that break them down into energy that your body can use. All right, no homework. See you all on Friday.